Today, it's my pleasure to talk with Kevin Tracey. He's the president and CEO of the Feinstein Institutes for Medical Research, where he's a professor at the Institute of Bioelectronic Medicine, which is the main focus of our podcast today, is this relatively new and rapidly developing field of trying to understand how one can modify the nervous system in ways that suppress inflammation and treat a variety of disorders. He's also a professor in the Zucker School of Medicine at Hofstra and the vice president of research for Northwell Health, which is a big healthcare network in the area up there in New York where he is. He's also editor, and I think founding editor of the journal Bioelectronic Medicine. Why don't you start, Kevin, by talking about your background? Where did you grow up? How did you get interested in medicine? And how did your career progress from being first a neurosurgeon to then hardcore immunologist and neuroscientist, and which is what we're going to talk about today.
Well, thank you, Mark. If you don't mind, I'd like to begin by thanking you for having me on your program. It's great to be here. I've been a huge fan of your work for decades, and I was really honored when you asked me to come on the podcast. So thank you for that. I grew up in the Northeast in Connecticut, and when I was very young, 5 years old, my mother died of a brain tumor, and her father was a pediatrician at Yale. Where she was operated on for a glioblastoma. And so from as long as I can remember, I remember my grandfather telling me stories of his work on polio vaccines at Yale, which was, of course, ground zero for the March of Dimes' first research efforts into polio. And he gave me actually a squirrel monkey skull from one of the monkeys that he had inoculated. This is how they discovered, the Yale team that he was a young investigator on, discovered the transmissibility of polio in sewage. And my grandfather wrote a paper in Science describing these findings. Charles Collada. So it's, for all my life, I've been exposed to neurosurgery and research. Now, is there a plan when you're 5 years old to become a neurosurgeon that does research? I don't know, Mark, but that's what I do now.
And that's interesting. You mentioned the polio virus in the sewage. We've seen that in the news recently.
It's incredibly important. There's a number of incredibly important parallels to the polio epidemics that swept through New York City, for instance. They originated in Pigtown, they called it, which was a home for poor Italian immigrants in that era, and the disease was blamed on the immigration problem, and you can look at the headlines from that era. Close the city, close the town. Stay off our trains. And having just sort of like relived it 100 years later, it's remarkable, Mark. And now tracking COVID in the sewage is happening in many major metropolitan areas around the world. And of course, there was just a recent confirmation of polio again in New York, and it was found first in upstate New York, and it was found first in the sewage. So yeah, at the time my grandfather did it. It was a fundamental piece of the puzzle, because the scientific dogma then, the accepted scientific model for the transmission of polio was actually respiratory, and the idea that polio could come in fecal oral meant it had to actually traverse the bloodstream before it could be systemic. And the team at Yale, led by Dorothy Horseman and others, was absolutely pivotal in enabling the development of vaccines by starting with understanding it was fecal oral.
Okay, and then when you went to college, you majored in chemistry. So, that's really if you know chemistry well, you're going to do really well in biomedical research.
Well, I agree. Chemistry is what is it? It's molecular tinker toys. You're taking things apart and putting them back together. And I worked in the laboratory of T. Ross Kelly, who was still alive. He's now semi-retired or emeritus, but he was a leader in natural product chemistry. And I'll never forget the first lecture I saw him give. He stood at the board and he sketched the structure of adriamycin. And he said, "This is the molecule that we're working on synthesizing in my lab." And my mouth dropped. And then he said, "This molecule is used to treat cancer." And I'm thinking, I'm a sophomore in college and taking organic chemistry, and I'm thinking, you can do that? You can use chemistry for something besides, what you're taught in high school in a chemistry book. You can build things that help people. And I said, I want in on that. And so I became a chemistry major. I worked in his lab for 2.5 years, and I worked on trying to synthesize a molecule vaguely related to adriamycin called bostricin, which was a red molecule that had been isolated from nature and was being tested as a chemotherapeutic agent. And I think I synthesized it, but I never got a positive NMR plot, so I couldn't prove that I had actually synthesized it. 2 or 3 years later, Ross and a graduate student published a paper that they completed the synthesis of bostricin. So my first contribution to the scientific literature is in the acknowledgement of Ross's paper saying that they had synthesized it. And thank you, Kevin Tracey, for doing some work a few years before that.
Well, that's good. And then you decided to go to medical school, where you went to Boston University Medical School.
The decision was hard for me, Mark. I really loved chemistry, and I wanted to be a medicinal chemist and make drugs. But I decided to go to medical school because I thought it would be the best way to invent things that would help patients, which was my dream all my life, to invent things that would help people. I thought the best way to do that would be to understand the patients and their diseases and to work from that perspective. And Ross Kelly begrudgingly blessed my decision, and I've stayed in touch with him ever since. But I think from interacting with patients at their bedside and from interacting with them in the operating room and seeing and understanding the things that go wrong, that provides not only insights that are very special, that are hard to get from not being a physician, but it also provides motive.
So you were interested in brain disorders. You mentioned your mother died when you were young of glioblastoma, a brain tumor. Did that have any impact on you deciding to do neurosurgery?
It probably did. Like I say, I'm very, as now that I'm 64, I'm very cautious of saying there was ever a plan when I was young. I think you get to be senior in your career and you look back and you assemble the story after the fact. I think the influence as a child, I was absolutely influenced by the knowledge of my mother's death, soon after a neurosurgery operation that was essentially useless. And I read lots of books in middle school and high school and college about neurosurgery. And I always, you try to, when you suffer a loss like that at that age, you try to figure it out in the ensuing years, what happened, what else could have been done. And so, I was quite sensitized to neurosurgery, but in medical school, I rotated on a neurosurgery rotation with Dr. Ed Spatz, and I was fascinated by the procedures of doing neurosurgery. And, he let me drill a hole into a human skull when I was a medical student. And that's quite unforgettable. And I always loved working in my father's workshop and now my own. I love woodworking and carpentry, and I love working on cars. So the whole mechanical aspects of surgery I was always fascinated by. And when the time came to choose a residency, I ultimately did choose neurosurgery for a number of reasons.
I went to high school with one of the kids in my class, which was in all my science classes. His name was Thor Sunt.
Oh my goodness, sure.
And
famous neurosurgeon.
Oh, his father.
His father, wow.
Yeah, his father is a famous neurosurgeon who ended up getting cancer. And I think they had this thing on 60 Minutes. They had a piece on him because he kept doing surgery when he couldn't even hardly stand up. Yeah, so that was up. I grew up kind of in an environment. I went to Mayo High School, although my father was actually a prosecuting attorney. But I'd say most of the kids in my science classes, their parent, at least one of their parents was a doctor at the Mayo Clinic.
Yeah, Dr. Sunt was a role model to generations of neurosurgeons.
Yeah. Okay, so let's get into it. And then probably your first really critical research, I think, from what I can tell, that led to your interest in neuroimmune actions was actually looking at the cytokine called tumor necrosis factor, or TNF, which is its levels increase dramatically when you have tissue inflammation. And that can be relatively localized to the site, say, of an injury, but then there are also these conditions that lead to like a systemic inflammation where you get this, people have termed it cytokine storm. And do you want to talk about your experience? I've watched a couple of your other podcasts and you always talk about your experience with this young, I think it was a young girl who died in your hands.
So yes, her name was Janice, and I was training at the New York hospital to be ultimately to be a neurosurgeon. And as part of my training, I rotated through the burn unit. And Janice was 11 months old, had been crawling across the kitchen floor when her grandmother was cooking dinner. And Janice, unbeknownst to the grandmother, had crawled between the grandmother's legs just as the woman turned to drain a pot of boiling water into the sink, and she tripped on the baby. And the boiling water scalded Janice over about 90% of her body. So she had essentially a lethal burn injury. Back in those days, her chances of survival were nil. But sort of despite the odds for 30 days and nights, she kept bouncing back. She had, we took her to the operating room for skin grafts on multiple occasions. We put her on and off ventilators, intubated her on multiple occasions. And, but despite the severity of these problems, she kept bouncing back, and she got to be very well known in the burn unit. Her face wasn't burned, and she was an adorable, angelic little girl. And so the whole place was rooting for her and to get better. And we thought she was going to go home, despite the odds. And then about a month into this, I was standing in the doorway to her room while she was sucking on a baby bottle, being rocked in the arms of a nurse at lunchtime. And as I stood there thinking, how nice it was to see this pseudo-normal scene, Janice, her eyes rolled back in her head. And I ran in the room and put her in the crook of my left arm, and I gave her CPR with my fingers and puffed air into her mouth while the nurse called the code. The whole hospital, it seemed, descended on the room, and this code went on forever. It felt like forever. We had cardiac surgeons there. We had pediatric anesthetists. We had respiratory therapists. Everybody was there. And but despite all this, she was gone, and it was horrible because she was so innocent, and it was such a surprise, and it was unforgettable. But to make it worse, we couldn't explain what she died of. She had gone into shock, and it really made no sense. That she had no evidence of any infection. We never found any infection, even at postmortem. When I got the autopsy results back, she didn't have any signs of infection. So the real question in addition to being haunted by having to tell her mother that her little girl had died, and watch her mother throw herself to the ground in a dead faint, I also had nightmares of not understanding what had happened. And so I was scheduled to go in the laboratory. This was at the end of May. I was scheduled to go in the laboratory in July with Tom Shires and Steve Lowry, two surgeons who were worldwide experts on shock. And I decided then and there that I would try to figure out what is it about septic shock? What causes it? How can it be treated? And that led to our early work on TNF as a mediator of shock.
Okay, what is TNF and what cells produce it?
So TNF stands for tumor necrosis factor, and it's produced by macrophages. And it's a molecule that is now would be called a cytokine, and it's actually a pivotal molecule in cytokine storm. When macrophages produce large amounts of TNF, TNF produces all the other cytokines that make up cytokine storm, like IL-1 and IL-6 and IL-8 and on down the list. But at the time, TNF was known by another name as well. So TNF had been discovered by Lloyd Old and his colleagues at Memorial Sloan Kettering, which was diagonally across the street from the burn unit at the New York Hospital. But in the same time frame, Tony Cerami and Bruce Beutler had discovered a molecule they called cachectin. And that was work was done at Rockefeller University, which was also on the diagonal from the other side of the street on York Avenue from the New York Hospital. So you had a triangle of these three institutions, the New York Hospital, Rockefeller, Memorial, where all this work happened. And so Tony had a graduate student or a medical student rotating through his lab. And Tony asked this student, did he know anybody at Cornell who knew anything about cachexia? Because Tony was studying this macrophage-derived protein that caused cachexia. And the medical student said, yeah, I just heard a lecture by this guy named Steve Lowry, a surgeon. So Tony called him and said, do you want to talk about cachexia? Steve gave me it was no voicemail back then. It was a handwritten note from the secretary, Judy. And I walked over to the Rockefeller University and introduced myself to Tony Cerami. Tony introduced me to Bruce. And the four of us with Steve began collaborating on what ultimately became an important discovery for therapy.
And you had your first key paper on that was with Tony, right? You had a Nature paper where you found that well, I guess you can describe what LPS is, but anyway, it's a molecule that's on bacteria and can induce this kind of cytokine storm.
Right. So the first paper I wrote on this topic was written to answer the question, if LPS activates macrophages to release TNF, is TNF sufficient to cause shock? And a paper that we published in Science with about 15 co-authors and I, including, as you said, Tony and Steve Lowry and Bruce Beutler and others, the first paper that I wrote said that yes, TNF is sufficient to put an animal into a state that is physiologically, pathologically, metabolically, essentially indistinguishable from lethal endotoxin poisoning. Now, endotoxin is made by bacteria, of course, but TNF is made by macrophages, by the immune system. And so this was a Science paper because it was a huge surprise that the body's own immune system would produce a molecule that could do this. And so that really led to the second paper, which was we published in Nature in 1987, which was again led by myself and at that point Bruce had moved to Texas, but Steve and Tony and I were still collaborating, and we used a monoclonal antibody against TNF. And here we asked the question: if you infect a baboon with bacteria, so that the bacteria stimulate the baboon's immune system to produce massive amounts of TNF, does monoclonal antibodies confer protection? And the answer was a resounding yes. And that turns out to be the first description of monoclonal anti-TNF antibodies as anti-inflammatory molecules. So that paper was very, very exciting and sort of put the stake in the ground that you could use monoclonal anti-TNF as a therapy.
And now these TNF antibodies are widely used for certain indications, rheumatoid arthritis, Crohn's disease, I think.
That's correct. And a number of other indications. I mean, they represent tens of billions of dollars of annual drug sales. And in fact, they were the first biologics in the anti-inflammatory biologic category of the pharmaceutical industry, which I saw a statistic the other day. It's projected that by 2025, this category of biologics will represent $325 billion in annual sales in the trillion-dollar pharmaceutical industry. It's unbelievable, actually. Yeah. It's amazing. It's amazing. And, it's helped a lot of people. These millions of people are treated with biologics for, as you said, rheumatoid arthritis, Crohn's disease, and other inflammatory and autoimmune conditions. But they're not perfect. They help about 40% of patients, and thankfully, that's a great thing. To benefit 40% of patients is great, but they don't benefit 60%, and they cost between $30,000 and $100,000 per year, and they all have black box warnings.
Yeah,
which means the side effects include cancer, sepsis, paradoxically, and immunosuppression, and even death. So alternatives are absolutely needed.
Yeah. And this is where you get into what's now called bioelectronic medicine. And
well, it also touches how we got there. Also touches your own work from your own lab, which you probably don't even know, but I'll tell you if you're interested.
Okay. I might guess. Does it have to do with like normal functions of TNF?
It has to do with TNF in the brain during a stroke.
Okay. Yeah, because you've said all this about TNF being a bad guy, but we did work a long time ago. It was actually, we got a result that was opposite of what we thought. We had cultured neurons in a dish. We'd been showing in the previous years that certain neurotrophic factors are good for the neurons. And then there was also this other literature on brain inflammation and Alzheimer's disease, activation of microglia, which is essentially the same as macrophages. And so I had a postdoc who I said, why don't you look at these pro-inflammatory cytokines and see if they're bad and see if we can protect the neurons with the good neurotrophic factors and so on. And lo and behold, he found if he pre-treated the neurons with TNF, they were more resistant to being killed, undergoing cell death, and a lot of adverse conditions. And that was about the same time that there's a number of different immunologists, actually some working on programmed cell death, including David Baltimore and others. So the idea was that the TNF activates a transcription factor called NF-kappa B, and that leads to downstream cascades that's bad for cells. But we and them pretty much at the same time found that actually activation of NF-kappa B is part of an adaptive stress response that's designed to keep the neurons alive or other cells alive.
I remember reading your papers very, very well and talking about them. And we had come to a very similar conclusion as you, because timing matters. So and dose matters. So it's absolutely clear today that as you led the way in thinking on much of this, that a small amount of TNF in the right place at the right time is good for you. And just as too much of TNF or TNF in the wrong place can be bad for you, or bad for the cells, or bad for the brain, or bad for the tissues. So 100% agree with you. You know, it's interesting. We had made a molecule that we called 1493, and the idea of this molecule, it's a long, complicated story that I won't get into, but my chemistry background clearly helped. But we were trying to make a molecule that would block the uptake of arginine into macrophages. And we screened molecules that had guanidinium groups that were arginine mimetics. And the idea was, if we could find a molecule that kept arginine out of the macrophage, maybe it would disrupt macrophage function and prevent excessive cytokine production. And the lead molecule in the screen that year was molecule number 14, and the year was 1992. But we didn't want to name it 1492 because everybody would talk about Columbus sailing the ocean blue. So we switched it, poetic license, to 1493. And that molecule we worked on for many, many years as a molecule to block TNF production and cytokine storm. And we were studying stroke in part because we had seen your papers. And we said, well, we could put the molecule in the brain and see what happens if we block not only TNF, but cytokine storm in the brain. Will that be good or bad during a stroke? And the answer was it was very, very protective. And that's what we expected. It blocked the cytokines. What we didn't expect is that when we put the small amounts of CNI 1493 into the brain to block cytokines in the brain, it also blocked TNF throughout the body. And that was the, you know, some people say the eureka moment. It's more like the holy crap moment. What is going on here? You know, that's what scientists really say, right? It's like, what is going on? How could this molecule in the brain block the cytokines, the TNF production in the spleen, in the heart, in the liver? You know, how did the brain tell the body to turn off TNF production? So that was the discovery that changed everything for me for the last 25 years or more.
And enter the vagus nerve.
Enter the vagus nerve. So for months in lab meetings, we would argue about these results. We repeated them over and over again. We knew it was true, but we said, well, I mean, it can't be coming out the spinal cord. That doesn't make any sense, the signal to turn cytokines off. Maybe it's the pituitary gland. Esther Sternberg and others had done a lot of work that the hypothalamic pituitary axis can turn off inflammation by activating the adrenal gland to make steroids. So we took out the pituitary glands of animals and we took out the adrenals and we repeated the experiment, and it still worked, meaning it wasn't the pituitary gland and it wasn't the adrenal. So then I came across a paper by Linda Watkins, a psychologist in Boulder, Colorado. And she had discovered that injecting IL-1 into mice and rats to disrupt thermogenesis in models of fever, that the signal required an intact vagus nerve. And so she had proposed that the vagus nerve was transmitting information about the presence of IL-1 in the abdomen up into the brain. And you know, so now I'm a neurosurgeon who knows about reflexes, and we know we've got this off switch that's activated in the brain. And I wondered if Linda's discovery that IL-1 signals up the vagus nerve was the on switch to a reflex. And so we cut the vagus nerve. And now when we repeated the experiment, the TNF was no longer blocked. And so at once we knew that the signal that CNI 1493 in the brain was mediated through the vagus nerve to control cytokines in the body. And that was an unbelievable idea, because we thought this is so simple, somebody must have thought of this. And we went in the literature and we could find nothing. We couldn't find anything about the idea of reflex control of immunity, and we could find nothing about vagus nerve controlling inflammation or cytokines.
No,
and so
we
started doing the experiments.
Of course, the early work on vagus nerve, you know, the dogma, the early work was all vagus slows down heart rate, right? That's the first finding. And then activate vagus, it will enhance gut motility. And, you know, there were at least two peripheral organs that it was known that it can affect, and I think maybe the pancreas, but
I think the list of vagus nerve reflexes, even in the 1990s when we started, was long. So there were, as you say, there were cardiac reflexes, there were respiratory reflexes, pancreatic reflexes, and gastrointestinal, and even some liver reflexes that had been sort of attributed to critical functional signals in the vagus nerve, and even the diving reflex. So these things were known, but the immune system was not on the list, and that was. But we said, thinking about it more, well, if all of these other organs evolved over time to have this intimate relationship with vagus nerve reflexes, perhaps the immune system was also exerting evolutionary pressure on the evolution of vagus nerve reflexes, and nobody ever looked before. So we started looking.
Yeah. And okay, so then the next question is, so you cut the vagus nerve and you no longer get peripheral inflammation when you cause damage or cytokine increase in the brain. How is that working? What is the vagus nerve innervating?
So that's actually still an area of active research. How the sensory information is transmitted in the vagus nerve, and what it does in the brain when it gets there is probably one of the hottest areas now in neuroimmunology to figure out. We have a paper that is soon to be in press. Perhaps by the time this podcast is out, it'll be in press. My colleagues, led by Sangeeta Shivan, my last author on many projects in the lab, and then grad student, then postdoc Harold Silverman, looked at this question as it relates specifically to IL-1. And what we've shown is that there's a family of nociceptors called TRPA1 nociceptors, which are defined as sensory neurons, A for afferent neurons that send signals up the vagus nerve. And these neurons express a cation channel called TRPA1 that's sensitive to cold, it's sensitive to reactive oxygen species and other chemical irritants, including cinnamaldehyde. And it turns out that what we showed is that when you selectively deplete TRPA1 from neurons, that mice become refractory to IL-1 signaling. So we also know the IL-1 receptor is required. So it appears as if co-signaling between IL-1 receptors and TRPA1 is required to mediate the vagus nerve dependent thermogenic responses to IL-1, which is really amazing.
Yeah.
And we found that those signals are also necessary and sufficient to turn on the motor signals in the vagus nerve, which return to the immune system to turn off cytokine storm. So the upgoing afferent signal in the vagus nerve converges on the nucleus tractus solitarius, and from there is relayed to other brain regions, including the dorsal motor nucleus of the vagus. The dorsal motor nucleus of the vagus sends cholinergic projections down to the celiac superior mesenteric ganglion complex, which in turn sends an off signal, if you will, a cytokine suppressing signal to macrophages in the spleen and other organs. So it's a fascinating and highly sophisticated reflex circuit that can be mapped in exquisite detail.
Right. So can you talk a little bit about, you talked a lot about macrophages, but there are other cells in the immune system, for example, T cells and B cells. Does the vagus nerve affect those subtypes of immune cells?
Yes, it does. And many groups are studying specific endpoints. I think a good way to think about this is that there's two vagus nerves, right? There's a right vagus nerve and a left vagus nerve. And each of them have about 80,000 fibers. So, you know, for those not familiar with neurophysiology, neuroscience in this space, when people say vagus nerve, they're actually referring to 160,000 vagus nerves. Yeah. And each of those fibers theoretically has a different responsibility, a different job description to control a different response. So we can walk and chew gum at the same time because our nervous system uses individual nerve fibers to do individual jobs. So when you start to break down what we know about signaling in the vagus nerve, for instance, Steve Lieberlis at Harvard, who's a real leader in the molecular and genetic mapping of vagus nerve functionality down to the single fiber level, Steve discovered that there's about 112 neurons in the vagus nerve that control respiration. I mean, that's a pretty small number.
Yeah.
And, you know, we estimate at most that 1,000 or between 500 and 1,000 TRPA1 nociceptors control the response to IL-1. And we estimate on the cholinergic side, the motor side, that probably less than 1,000 and probably again in the hundreds of fibers are required or necessary to turn off TNF production in the spleen in a mouse during endotoxemia. But notice that I added another qualifier there, right? So as you start to ask questions about functions, the functions are going to be contextually dependent on what else is going on. Is it a model of endotoxemia? Is it a model of arthritis? Is it a model of colitis? Where are the cells that you're interested in? How do the nerve fibers get there? Where do the nerve fibers terminate? And so we are literally just beginning these kinds of explorations. You know, you can make some generalizations, right? The vagus nerve cholinergic fibers terminate in the ganglia, which is the origin of the splenic nerve. The splenic nerve is catecholaminergic. The catecholaminergic splenic nerve releases norepinephrine into the spleen, which is like a venous lake. And so now you're not looking at sort of a nerve ending specific effect on all the T cells, which you mentioned, and the B cells, which are in the spleen, but you're looking on at more of a humoral neurotransmitter process. So these are burning research questions now, but the importance of your question is you can ask it. Yeah. You can say, I'm interested in T cells or B cells or dendritic cells, and these are the neurons in the region of the cells I'm interested in, and these are the neurotransmitters they produce. So what is the effect of these neural signals and these neurotransmitters on this specific cellular response of this cellular subtype? You can do the experiments now with genetics and frankly, with nerve stimulating and nerve recording devices that weren't available even 10 years ago.
Yeah, it's very exciting from a basic research standpoint. You know, work done in animals, which generally, when you're trying to understand the details of mechanisms, you start with animals. And so while that's going on and you're continuing, you know, trying to discover those kinds of basic neurochemical, anatomical interactions between the nervous and immune systems. In parallel, you've been doing a lot of work on clinical applications of vagus nerve stimulation, and you've had some exciting success in early studies, and you're involved either directly or indirectly in a number of other clinical trials. So do you want to start off talking about, I guess, actually epilepsy, which I guess wasn't your work, but it was one of the first, maybe the first application of vagus nerve stimulation that has approval by the FDA.
Yes, so you're absolutely right. I mean, the science begins in the lab with mice, and the advantage to the mouse models is that we can use very sophisticated genetic manipulation to isolate and identify very discrete mechanisms. We use we use optogenetics. We use now a technology called trapping, so we can identify specific brain neurons that are activated during specific inflammatory conditions, and then we can go back and reactivate them to mediate specific inflammatory changes in the body. So the power of the discovery science in mice is incredible, and it's incredibly important because you really need to know what you're doing mechanistically before you try to move into patients. Yeah. And in the device industry, that wasn't always the case. And that's in fact the case with epilepsy. Vagus nerve stimulation was first approved by the FDA for use in treating drug-resistant epilepsy. I believe it was 1997. I might be off by a couple of years, but long time ago, the FDA granted approval to implant vagus nerve stimulators in humans to treat epilepsy. What is the mechanism? No one knows then. No one knows today. And as a result, you have a therapy that has been used, probably about 200,000 patients worldwide in the last 25 years have been treated with vagus nerve stimulation for epilepsy. But like the biologics for autoimmune disease, it only works in about half the cases. So up to 60% of people with vagus nerve implants for epilepsy, they end up getting it, just turning it off, because it doesn't do anything. Now, fortunately, it helps 40% of the patients, and that's great. But no one knows why it helps the patients it helps and doesn't help the patients it doesn't help. So that it wasn't mechanism based. Yeah. However, the good news is, to the extent that we can learn from implanting vagus nerve stimulators in humans for 25 years, that it's safe, it can be safely done. It is not immunosuppressive. There's no black box warning. And it doesn't cost $100,000 every year like biologics do. It costs whatever, 30, 50. If it did cost $100,000, I don't think it does. It's once. It's not once every year. So this has laid an important groundwork that although it is an invasive technology, some would argue injecting biologics once a month is also invasive, and that it's time to look at what is the clinical evidence that the advantages of a once and done surgical implant of a vagus nerve stimulator may outweigh the risks of biologics for a lifetime. Yeah. And so those are the kinds of questions that are happening now. I co-founded a company called SetPoint Medical. It's a Valencia, California company that's now been in operation for 15 years. They've completed several clinical trials for a vagus nerve stimulator that's the size of the tip of your little fingernail. It's the size or smaller than the fish oil pill you may have taken this morning. It gets implanted in the neck. It's been implanted in the necks of patients with rheumatoid arthritis, and the FDA recently authorized breakthrough designation for this therapy. And the first arm of the clinical trial was completed a few months ago, and a couple of weeks ago, the FDA authorized the second arm, which I believe the first arm was 60 patients, and the second arm is going to be 190 patients. And this is very, very exciting because there were no changes or modifications made after the data was looked at for the clinical trial. So time will tell. I estimate there's been about 100 patients treated worldwide with rheumatoid arthritis and Crohn's disease with some form of implanted vagus nerve stimulation or other. My colleagues and I at SetPoint published in the Proceedings of the National Academy of Science a paper from a study led by Paul Peter Tack and his student Frieda Koopman, which looked at patients with severe rheumatoid arthritis, and the results were highly statistically significant, showing benefit. So yeah, the story continues. It takes a long time to go from mouse to humans, but this story continues to progress in good speed.
And what are some of the other diseases or conditions that there's currently clinical trials?
So for vagus nerve stimulation by surgical implants, I'm aware of clinical trials that are either have been done or are being done for rheumatoid arthritis and Crohn's disease.
Okay.
But when you move to non-invasive strategies to stimulate the vagus nerve, the list grows exponentially. So ultrasound can be harnessed to stimulate the vagus nerve in the porta hepatis of the liver or in the spleen. And there are ongoing clinical trials for rheumatoid arthritis, metabolic syndrome, obesity, diabetes, using that modality. And then when you move to non-invasive stimulation of the nerves in the ear, which is called transauricular vagus nerve stimulation, which I argue may or may not be a misnomer. But some of these devices have received FDA approval for migraine headache, for rehabilitation therapy after stroke, and for treatment of opioid withdrawal. So the list is rapidly growing for invasive and non-invasive indications for direct vagus nerve stimulation, which would be, of course, the surgical implants. That's where you really know exactly what you're doing. And non-invasive, which there's growing interest in.
So with there's disadvantages and advantages to both approaches, right? For non-invasive, the advantage is you don't have to have any surgery. The disadvantage is you have to be able to stimulate, I guess, every day or, certain intervals. And so you have to think about it and do it. And, over periods of months and years, that could occupy a lot of time and effort. Is that kind of and also, as you mentioned, with the implant, you're stimulating exactly where you want to. And whereas with external, probably the current, depending on where you put these external electrodes from day to day, it might even change how much the actual nerve is being stimulated.
That's right, Mark. I think, look, all of this is going to be subject to a massive cultural change in medicine and in the pharmaceutical and device industry. And if you start with, from the patient's perspective, what could be easier than taking a Z-Pak, right? Where it's one antibiotic a day for 5 days. Well, the data are patients don't take all 5. What could be easier than taking 28 amoxicillin in 14 days? The data are patients don't take all 28 pills in 14 days. Mm-hmm. Compliance is a huge issue in medicine. So the trade-offs that you talk about, surgical, non-surgical, invasive, non-invasive, once a day, twice a day, 4 times a day, it's going to be, I think, what we're going to live through in the coming years as these therapies find their way into mainstream and widespread use. And I think they are destined, frankly, to find their way into mainstream widespread use. It's going to be very disruptive. And I think it's going to be figured out, where we end up will be figured out disease by disease and severity by severity. So for a serious disease that can land you, in an operating room or in a hospital, or frankly can kill you, if the option's available to have a one and done implant that's 30 minutes, local anesthesia, in and out in a few hours, and then forget about it for the rest of your life, except for your every month or two, every 2-month doctor visits to have it adjusted or tuned by the doctor's iPad. that's going to be a very attractive option for some people who are currently going to the doctors once a month to get injections of a drug that has a black box warning and costs $100,000 a year and doesn't work half the time.
Yeah.
So on the other hand, you've got some transient problem that may or may not be treatable with a handheld device that the patient might find fun to do. There'll be a role for that. And then in between is going to be a chronic condition that, diabetes management, for instance, glucose management, which should be carefully monitored and carefully done, but could be done by an ultrasound or a handheld device of some other kind. I think it's going to depend on the patient and the physician. Yeah. You have a highly compliant patient who really wants to take things into their own hands. They might want to do it themselves. Another patient who's too busy to follow this thing all day long. They're going to say, plug me in with a small device that I can forget about for the rest of my life and take care of, let the device and the computer and the AI and the machine learning and the biofeedback into the cloud take care of my glucose. Do it. So those are the kinds of choices I think that are, like I say, I don't think this is, to me, that there's so much data now in human data and laboratory data and mechanism data and genetic data and mouse data, to me, that the rocket ship is launched and we're not making predictions about the future anymore. We're just tracking the trajectory of the rocket.
Now, there's certain lifestyle factors that, at least from the standpoint of the efferent signals going down the vagus state of the heart or the gut, regular aerobic exercise, we've done work on intermittent fasting, they tend to enhance parasympathetic tone. And I guess, did we even mention that? Yeah, so the vagus is part of the information coming down is called parasympathetic, and that slows heart rate, enhances gut motility. So I'm wondering to what extent, electrical stimulation of the vagus nerve is mimicking to some extent, or at least through that mechanism, some of the beneficial effects of these lifestyle factors.
Yeah, that's a really important question, and the answer is no one knows. Yeah. I think we should talk about this for a minute. I think we should also talk about the implications for vagus nerve damage, which has been talked about a lot recently in the context of COVID. So to talk about the relationship of vagus nerve health to blocking inflammation, to general health, there's at the mechanistic, at the physiologically verifiable level, factors that increase vagus nerve signaling to the heart, meaning slow the heart, tend to be associated with health and decreased inflammation. So whether it's, as you said, intermittent fasting, cold ice baths, cold showers, chronic aerobic exercise, even fish oil consumption and meditation, cognitive behavioral therapy, and singing in a choir and prayer, all of these things increase heart rate variability, slow the heart by stimulating the vagus nerve. Does that mean that is why they are anti-inflammatory? We don't know. It probably, but as I said before, it's not a solid copper wire, the vagus nerve. It's 160,000 fibers, and so you can certainly believe or know that the fibers to the heart that slow it down they're not necessarily the same fibers. They're probably not the same fibers that go to the spleen or go to other organs to control inflammation. Yeah. And I think the evidence that they all fire at the same time is just frankly wrong. There's this idea that vagus nerve control centers fire en masse. I mean, that makes no sense. No. Steve Lieberlis has gone a long way to disprove that, frankly, with his 112 neurons that control breathing in
Well,
different synchrony.
that'd be like thinking that when your sciatic nerve is normally activated, it's activating all. If that was the case, you'd just have, contraction of all the muscles and from your big toe all the way up to your hip. So
You'd have tetanus in your leg.
yeah, that's right.
So I agree. I think it's fascinating that that all of these things that are generally associated with health tend to slow the heart, which is a vagus nerve stimulated activity. But as a scientist or as a physician, we don't know yet what the relationship of the controllers is to the various outputs. And but you can study it, right?
Yeah.
You can, and we are studying it in mice. It's much easier to study it in mice because you can tag the neurons, you can record the neurons, you can reactivate the neurons after various interventions. It's a lot harder to study in people, but there's some interesting correlates to this in people. So the one that got the most attention in the last 6 months or a year was for long COVID, because a fascinating study came out of, I believe it was Spain or Portugal, Madrid, I guess, looking at ultrasound analysis of long COVID patients and imaging their vagus nerve in the neck. And what they showed is a lot of these patients had severe damage to their vagus nerve. Wow. And that's fascinating, because a damage to the vagus nerve would be like, in the context of cytokine storm or inflammation, would be like damage to the brake lines in your car. So if you're going down a mountain, you want to make sure your brake lines are working properly. And if the brakes are damaged in a COVID patient, you're going to be a setup. Those patients could theoretically be a setup to have unregulated cytokine release and unregulated inflammation chronically. And so if that's the case, it hasn't been proved as a cause and effect mechanism yet, but it's very possible that the damage to the vagus nerve contributes to the uncontrolled or ongoing inflammation in some patients with long COVID. Now I have to be really emphasize here. This doesn't mean that acupuncture and meditation and ice baths are always going to stimulate your vagus nerve, and that patients should take this into their own hands. If the vagus nerve is damaged, those modalities may not be effective. Yeah. And the fact that you can see the damage with an ultrasound imaging in some patients is interesting, sort of proof that the vagus nerve can be damaged. But there may be other types of vagus nerve injury that aren't going to show up on an ultrasound, or could be very hard to diagnose. And so I get very frustrated when I look on YouTube or in social media and I see various people advocating, well, just stimulate your vagus nerve this way and you'll be fine. Maybe some of these modalities would help some patients, but it's not fair to the patients to say that these modalities will always be helpful, because we just don't know enough yet.
Yeah. And what about effects? So I saw this paper, recent issue of Neuron, which is kind of a top journal in the neuroscience field. Vagus nerve stimulation drives selective circuit modulation through cholinergic reinforcement, but they found in mice that vagus nerve stimulation enhances skilled motor learning. So it enhances the ability of these mice to learn a certain, moving their paw in a certain place, essentially learning to move their paw in a certain place. And that's mediated by acetylcholine signaling, which I think you're also finding in the spleen, right? That there's a role for acetylcholine. So that's kind of interesting. This, of course, they didn't really work out. Oh, they do think it's affecting what's going on actually in the motor cortex. They did add some evidence for changes in the motor cortex.
Yeah, it's a fascinating study, and it's an important study, because it puts into context a couple of things that have a background that goes back many years. So many years ago, I believe it was a paper in Nature Neuroscience or Nature. Investigators took subjects who had vagus nerve stimulators implanted, but had them turned off. As I said before, many subjects with epilepsy, the treatment doesn't work, and they just turn it off. So they brought them in to a study center, and they gave them a mental task before and after they turned the vagus nerve stimulator on. And they showed that turning on the vagus nerve stimulator. Now, when you stimulate the vagus nerve with these devices that are commercially out there for epilepsy, the lead is an alpha helical coil that's wrapped around the whole vagus nerve. So when you stimulate the nerve, you're stimulating the nerve up into the brain and down into the body. And what they found is when they turned the device on, the patients were able to complete the cognitive tasks much better, highly statistically significant improvement of learning and memory functions. And in subsequent work done by many groups, there's evidence that stimulating the sensory vagus nerve enhances hippocampal function and learning and motor tasks. So this was the basis for the strategy for groups to look at vagus nerve stimulation to enhance rehabilitation after stroke by pairing vagus nerve stimulation with motor learning. So there's a tremendous amount, and this is FDA approved now in the US. So there's tremendous amount of interest in how sensory vagus nerve signals enhance learning and memory and motor function. And there's also a tremendous interest in understanding how motor vagus nerve signals going down the vagus nerve control inflammation, glucose homeostasis, GI function, and even the gut microbiome. So it's a very, to the extent now that we're just beginning to understand some of these mechanisms, where we're headed is to a scenario where sort of individual groups of neurons are identified as the basis of highly specific circuits that control specific mechanisms in these various conditions. But we're really at the beginning of all this.
Yeah. Okay, let's see if I had any other things. I jotted down a few things. You mentioned evolution early on, right? So, what's the importance of this inflammatory or anti-inflammatory reflex from an evolutionary perspective? And since it's turning out that these nerve fibers going through the vagus nerve, some of them seem to be important for controlling, say, one organ, or maybe whatever, the spleen, and then you've got these other ones going up to the brain, and maybe affecting things like excitability of neurons. Epilepsy, somehow they're suppressing excitability, or maybe enhancing synaptic functions involved with learning and memory. So can you think of any, so what were kind of the environmental challenges that animals had and their human ancestors? And have you thought a lot about that?
We have thought a lot about that, because it's really important, I think, to understand how the mammalian nervous system emerged from evolutionary history, started from much simpler animals. And when we were working on this a long time ago in the 1990s, soon after we proposed that vagus nerve reflexes control inflammation, we were thrilled to see a seminal series of papers from Alejandro Abele, which mapped comparable reflexes in C. elegans. So C. elegans is a primitive worm, and what Alejandro and his colleagues discovered is that what I would call the C. elegans vagus nerve, and I'm taking poetic license there. C. elegans doesn't really have a vagus nerve; it only has a few nerves actually. But there's a set of cholinergic neurons in C. elegans that is activated by the presence of pathogens. Oh. And when the C. elegans is exposed to a pathogen, these neurons get activated. And activating these neurons in C. elegans is a way of downregulating the macrophages, the primitive immune system, in the same worm. So here you have an evolutionarily ancient roundworm, arguably one of the most ancient animals that we study today, that has essentially an intact inflammatory reflex. So we call it the inflammatory reflex because it's activated by inflammation, and it activates reflex circuits that, as you point out, are anti-inflammatory, but it also probably activates other responses as well that are yet to be named and yet to be studied. And so we've because science, reductionist science, our modern scientific world that you and I grew up in and live in today is a world of reductionist science, meaning in order to convince the world that we understand the mechanism, we have to keep reducing it to the next simplest first principle. And prove, if it's the vagus nerve, then what neurons is it? Well, it's the cholinergic nerves. Well, if it's the cholinergic nerves, where do they originate? Well, they originate in the dorsal motor nucleus. If they originate there, where do they terminate? Well, they terminate in the celiac superior mesenteric ganglion complex. Well, if they terminate there, what do they signal on? Well, they signal on the origin of the splenic nerve. And you keep going down the funnel until you get to acetylcholine in the spleen being released by T cells that express cholineacetyltransferase, the rate-limiting enzyme in the biosynthesis of acetylcholine. So it's a T cell subset at the end of the line that's making acetylcholine. And so that's how you do the reductionist first principle approach to mammalian physiology. And when you go the other way, you go backwards in evolutionary time, you converge on the roundworm that has a similar cholinergic reflex that controls inflammation in response to pathogens. So it's a nice story.
Yeah, and it also makes a lot of sense, as you know. So you just mentioned the T cells produce acetylcholine, which was first discovered as a neurotransmitter. On the other hand, the immunologists discovered all these cytokines, but now we know actually neurons not only can respond, but I think they can produce, at least some cells in the brain can produce some of these cytokines.
Absolutely. And, peripheral neurons can produce cytokines too. So a year or two ago, my colleague Juan Yang and Sangeeta Shivan and others, we published a paper in the PNAS, which was fascinating in this context. So what we found is if you put a ligature on the sciatic nerve, you produce an animal model of sciatica, which is like an animal model for when a herniated disc produces sciatica in a human. And like in the human, what happens to the animal with sciatica, the foot and calf get to be very painful. And you can measure this with a von Fries filament or a pain sensitivity test in mice and rats. And what we found was that what happens is that the inflammation in the distal extremity of the rodent is caused because the nerve endings release a cytokine called HMGB1. And we show that if you block the release of HMGB1, the animal doesn't have pain and doesn't have inflammation. So it's not the ligature on the nerve that's causing the pain. It's not the herniated disc, I believe, in the back of the human. And I'm a neurosurgeon that operated on thousands of herniated discs. I don't think it's the inflammation in the nerve at the level of the disc that's causing the problem. It's the fact that the nerve endings are releasing inflammatory mediators, and the release of the inflammatory mediators downstream in the tissue sends signals back up different nociceptive fibers, and that's where the pain is coming from. Huh. So it's a new way of thinking about this relationship of what is a cytokine, what is a neurotransmitter, and how do these things come together? It's a fascinating new model.
And so inhibitors of HMGB1 being identified or developed for pain conditions?
So we've developed, so they're not in clinical use, but many have been developed. We developed monoclonal antibodies that were effective in this model. We've also, working with our chemist and our co-director of bioelectronic medicine at the Feinstein Institute, Yusuf Alabed, we've also made peptides that selectively compete for binding with HMGB1 and its receptors. And this also prevents the pain in these animals. So there is a potential path forward to make HMGB1 antagonists in the treatment of pain, but it's a new field. But I'm just fascinated by the fact that the origin of the HMGB1 is the neurons themselves in the tissues.
Yeah.
So the nerves are not only, some of the nerves are transmitting pain signals back to the brain, surely, but other nerves are actually producing the mediators that are causing the pain.
You know, getting way back in evolution. So, I mentioned that immunologists were surprised to find that neurons produce certain cytokines, and neuroscientists were surprised to find immunologists found neurotransmitters being produced by immune cells. So I've thought about, which came first, organisms with nervous systems or organisms with immune systems?
What do you think? They came at the same time.
Yeah.
So Max Cooper has studied this, and it's interesting that animals evolved from sort of jellyfish-like neural network models to animals with primitive brains in the same time frame that memory T and B cells evolved. So I've talked to Clifford Wolf about this a few times, the neuroscientist at Harvard. And he and I are in agreement that the nervous system and the immune system are the same thing.
Okay, that's a good place to end the podcast. I very much enjoyed chatting with you, and I appreciate you taking the time, Kevin. And this is an exciting area. I'm going to follow it for my own reasons. And no, it's very exciting that there's these developing safe approaches that have a lot of potential for helping a lot of people who need help.
Well, it's great talking to you, Mark, and thanks for having me on. I really appreciate it.
Okay, bye.